So Konstantin actually is now a postdoc here in Uppsala in my group, but today is going to present the work that he did during his PhD in the group of Martina Dvořáková in the Czech Republic in Brno. So it's going to tell us about histone variants and repetitive regions in the Arabidopsis genome. So, floor is yours.
Okay, so thank you, Stephanie, for the introduction, and thank you to the organizers for the possibility to present my work. This work was called Analysis of Histone Variance Distribution in the Repetitive Regions of Arabidopsis thaliana genome, with main focus on H3 histone variants and RDNA as a repetitive region. So a brief introduction to the RDNA and to the nucleolus. So RDNA is transcribed within the nucleolus by RNA polymerase 1, and it is transcribed into the primary transcript that is further processed in 3 parts that afterwards assemble with ribosomal proteins and give rise to the ribosomal subunits that are further exported outside of the nucleolus. There are 750 copies of RDNA per haploid genome, so its amount is excessive. And basically, the main motivation for any study of the nucleolus is whenever you need to synthesize any protein, you need ribosomes. So it's important to understand how they are, how the RDNA repeats are regulated. So as I mentioned, this RDNA amount is excessive. Regulation is achieved by several epigenetic mechanisms. It is achieved on the level of DNA methylation, basically by methylation of the promoter. Also, it can be done by chromatin regulation, by histone variants and histone chaperone, histone modification, sorry. And also RDNA is kind of regulated at the chromosomal level. It was shown recently that active subtypes of RDNA are localized at one chromosome, while the inactive ones localize at the other one. So the main problem with any study of RDNA is presence of these active and inactive repeats. And we were aiming in our study to understand how the active and inactive copies are recognized or distinguished. So we planned to circumvent this problem of repetitiveness by using low copy lines that were previously obtained in our laboratory, where only active copies of RDNA are left. So also a brief introduction to H3 variants. H31 and H33 histone variants, they differ just in 4 amino acids, but they have different deposition dynamics, where one H31 is associated with S phase, and H33 is a kind of replication independent variant. Then they have also different chaperones associated. H31 has CAF, and the H33 has HERA and ATRX. And also they have different deposition pattern, where H33 is more associated with L-chromatin, and H31 with heterochromatin. And on the scale of a gene, H33 also has a distinct pattern from H31. For example, it is enriched at the 3' end of RNA-pol2 transcribed genes. The differences between these 2 histone variants, they're also detectable using microscopy. So you can see that H31 is abundant in chromocenters and in the nucleoplasm, while H33 was not found in the chromocenters. But it was found to form these condensed foci within the nucleolus that are dissociated after RNA polymerase 1 inhibitor treatment. So there's some link that was further reported between H33 and RNA polymerase 1 transcription within the nucleolus. So this led us to this study to kind of investigate how H31 and H33 participate in regulation of the RDNA. So we begin with some simple observations of wild-type line with a regular RDNA amount and a low copy line with reduced RDNA amount that carried this H33 GFP construct. So we found that in wild-type line, we could easily identify condensed foci stained with H33 within the nucleolus, which is shown here on this image by these arrows. And we could not find these foci as abundant in the low copy lines, as you can see from this image. And also, when we performed FISH on roots of these low copy lines and wild-type lines, we found that RDNA signal mainly comes from within the nucleolus, while in wild-type it also comes from within the nucleolus, but also it forms these condensed foci on the periphery. So this data showed us that there is like a link between H33 foci formation and RDNA amount. Next, we also had a look on the deposition of H31 using a line with a wild-type RDNA amount that expressed H31 and H33 simultaneously, and they were labeled with different fluorophores. So we analyzed these condensed foci within the nucleoli, and we found that most of our foci are stained with both histone variants. After that, we asked ourselves if these variants, they create some kind of distinct environment within these foci within the nucleolus. And if this environment is distinct from the rest of the nucleoplasm. So to analyze that, we employed this flame thread technique that allows to detect interactions between proteins at a very close distance. And we analyzed this experimental line with wild-type RDNA amount that has these condensed foci, and the control line that was carrying just the H31 GFP construct. So here at the results, we can see that on this image, the control in the control line, the lifetime was quite similar between individual nuclei. And it was also kind of homogeneous. You can see that they all appear bluish. Well, in our experimental line, you can see that the lifetime differed because the nuclei appear more greenish, and also the lifetime differs between the individual nuclei. And data is also presented in form of this graph, where you can see that control lifetimes, they can be fitted within this nice box plot, while this red-green plant, the experimental line, the lifetimes were quite different according to the amount of donor and acceptor dyes, but they were reliably different from the control. So here with this data, we identified that H31 and H33 are positioned in close proximity within this foci, and this pattern is not very different from the rest of the nucleoplasm. And here with this data from simultaneous immunodetection and FISH, we show more directly that H31 and H33 are associated with RDNA. As you can see here, it is indicated by these arrows, the places of colocalization. We also used some bioinformatics methods to analyze RDNA. So here, first of all, about the data representation. Here is a ChIP-seq graph for the corresponding histone variant. And here is a scheme of the RDNA repeat with all the basic components of it. And also in the data here, you can see these allometer regions. So we excluded them from the analysis we analyzed them by a certain algorithm. We found that they are very repetitive, and therefore they are not reliable, and we excluded them from the analysis. So here we took a public data set from this Wallman article comparing H31 and H33 distribution in dividing and non-dividing tissues of Arabidopsis thaliana. And we found that in dividing tissues, the amount of H31 and H33 RDNA was quite similar. And the only distinct feature was this small H33 peak at the transcriptional start site. Well, in non-dividing tissues, the levels of H33, they stayed quite the same, but H31 was much more prevalent and its amount increased. Also, we performed some ChIP-seq analysis on our own lines. So here we compare H3 distribution in wild type and low copy lines, and we can see that the patterns of distribution are quite similar, although the levels are different. And basically this conserved pattern, I think it shows that there is no major kind of rearrangement of H3 at the locus that occurs with the activation of the repeat. We also analyzed some epigenetic modifications, both bioinformatically and by this dot blot analysis, like ChIP with consequent dot blot detection of various positions along the RDNA locus. And we found that these two marks, H3K4 and H3K27, their amount is quite small at the RDNA. And basically, it was homogeneous between the analyzed lines. While the main difference was observed for this inactive modification of H3K9 demethylation. It was present in the wild type, but it was like largely depleted in the low copy lines. And here also with microscopy, we confirm some of this data by identifying H3K9 within these condensed foci in the nucleolus. And we also show that H3K4 and H3K27 are not really found within the nucleolus. So to summarize some key findings, condensed foci formation within the nucleolus is associated with RDNA amount. Both H31 and H33 are found in the condensed foci. H33 distribution pattern at RDNA differs from one and are unable to transcribe genes. H31 to H33 ratio changes are different at RDNA from the rest of the genome. Epigenetic modifications associated with constitutive heterochromatin were prevalent at RDNA. H3K9 was identified in these condensed foci. And these condensed foci, they can be basically characterized by an unusual combination of marks, such as H31, H33, and H3K9. So thank you for your attention. And if you got interested, you can find more information in this article.
Thank you very much, Konstantin, for this detailed talk about RDNA. I have to say, I have just learned that there were 2 chat boxes. So there were, in fact, many questions to Sarah before, and I apologize. But now I can see your questions, in fact. So if you have questions, please go ahead and type in. This time, I hopefully will see them. So I can start just asking a general question. What do you think? I mean, you see all these differences in terms of chromatin enrichment, different variants, etc., between RDNA genes and the rest of the genome. What do you think is really particular about RDNA transcription that makes it need to be different from transcription of other genes?
Yeah, so basically this, I think it's just the simultaneous need for both active and inactive copies. Yeah, so this basically is the thing in the background of it, I would say so.
So they have to switch more.
They have to be dynamically regulated. So there is this subset that seems to be constitutively silenced, or it is active. Like all of the RDNA is active at some of the very early points of the development of the plant, and then it kind of is divided into these two subsets. One that is completely inactive, and the other one that is active. But with this one that is active, it's also sometimes inactivated and sometimes activated, just to kind of adjust the cells to the basically things that are happening in the environment.
I see. So in fact, when you're doing your analysis and you see certain enrichments for certain chromatin marks, you don't know whether you're picking up from an active or an inactive specifically.
Yeah, with this, sometimes we do, sometimes we don't. So that is kind of what was a bit complicated in presenting this work. So for example, when we analyze here, we analyze also low copy lines, and it appears like all the RDNA repeats within these low copy lines, they are active.
Yeah.
So here we are kind of focusing on these active ones.
Okay.
But here in case of wild-type line, we are focusing on both active and inactive. So here, for example, this data, yeah, this one. So this comes both from active and inactive repeats.
Okay. Yeah. I don't know if you have, Sarah, you have any question? Or?
Yeah, sorry. It's indeed about this data about the different levels of histone 3.3 and 3.1 that you say that it's between highly dividing tissues. Yeah, so this is a link to the RNA polymerase. So the activity of.
Yeah, so this is like that. This data set from Wallman, it has data from young leaves and from the older ones.
Okay, so it's not meristematic tissue. So I don't know why I assumed that it was meristematic. Okay, so it's young leaves and older leaves, just to confirm.
Yeah, and so the young leaves, they kind of represent these dividing tissues, and the older ones, the non-dividing.
Okay. Thank you.
And basically the assumption is that in dividing tissues, there will be more of these active RDNA repeats, because the cells, basically the organ, the leaf needs to expand. So therefore, there should be some differences, and we indeed detect some.
Okay. Then I think we are just on time. Thank you very much for your nice talks. I'm sorry, Sarah. I think, can you see the questions here on the left?
Yeah, I think that some of them were already commented.
Okay,
no?
Yeah, some of them, yes. So I think now we should switch off this room, and then in 20 minutes, I think we have another session from the InDepth Academy starting. So thank you again, Sarah and Konstantin, and see you around.